In this video segment, we will begin to discuss pharmacokinetic principles and cover the topics of absorption and routes of administration. The learning objectives for this video are to understand the advantages and disadvantages to the different routes of administration, to know what bioavailability is and reasons for poor bioavailability, and to understand what is meant by the first pass effect. First, we'll start with a few key points for understanding absorption and routes of administration. Absorption is the transfer of drug from its site of administration into the bloodstream. The barriers that must be crossed to reach the bloodstream differ by route of administration, and the route of administration influences how quickly a drug will be absorbed into the bloodstream and how much of the drug will be absorbed and reach the target site. Drugs can be administered through a variety of routes. The body does not have a specific system for the purpose of absorbing drugs or toxicants. Absorption occurs through the same processes that are used for the absorption of essential substances, such as food, nutrients, and oxygen. For most drugs, the goal of administration is to reach the bloodstream, where they will be transferred to the target site. However, there are some exceptions. For example, when the target of the drug is on a specific part of the body that doesn't need to be accessed by the blood, such as the skin or within the GI tract. There are two main terms that are used to describe routes of administration. Enteral refers to administration via the GI tract, mouth, or rectum, and parenteral refers to administration through a route other than the GI tract. In this course, we will discuss the most common routes of administration. Oral administration is the most common and most preferred for drugs. Intravenous is directly into the vein by injection. Intramuscular is into the muscle by injection. Subcutaneous is an injection into the subcutaneous layer under the skin, and intradermal is an injection into the skin layer. Dermal or topical refers to drugs administered on the skin, usually in the form of a gel or a patch. Intrarespiratory refers to drugs and toxicants that are inhaled. And intraperitoneal administration means it is administered into the body cavity. This route is typically used in research, but is also the preferred route for some cancer chemotherapeutics. This figure illustrates what the four main routes of administration done by injection would look like. Now we will go through each of the routes of administration and discuss how drugs and toxicants are absorbed from each of these different sites of administration. Anything administered intravenously does not need to be absorbed because it is immediately present in the bloodstream. This is the quickest way to administer a drug. Administering a drug IV also ensures that the exact dose will be present in the blood. When a drug is administered IM or sub-Q, it must diffuse from the site of injection in the tissue into the bloodstream. Typically, these forms of administration create a slow and sustained absorption process. IM injections are absorbed into the blood more quickly than sub-Q injections because muscle tissue is more vascularized. Drugs or toxicants that are administered transdermally must diffuse across the skin layers and into the bloodstream. The more lipophilic the chemical compound, the more easily it will diffuse through the skin layers. Drugs that are given by inhalation are absorbed by capillaries in alveolar lung tissue. To get into the bloodstream from the alveolar space, they must diffuse across the alveolar epithelial cells and the capillary endothelial cells to enter the bloodstream. In addition to drugs, toxicants that are inhaled can enter the body this way as well. The oral route is the most common type of administration of most drugs and exposure route for toxicants. Drugs taken orally can be absorbed at any point of the GI tract. For a drug or toxicant to be absorbed from the GI tract, it must pass multiple layers. From the intestinal lumen, a chemical must cross mucus and water layers, then the epithelial cell layer, the basement membrane, and the capillary endothelial layer. In order for a chemical to be absorbed into the capillary, it must be capable of crossing the membranes of the cells that make up the epithelial layer and the capillary endothelial layer. To do this, they must either be lipophilic enough to diffuse across the membranes, or utilize transporters that will allow polar or charged molecules to cross these biological membranes. A number of factors influence the absorption of drugs and toxicants from the different regions of the GI tract, including lipid solubility, ionization, pH, the presence of transporters, surface area, stomach contents, and first-pass metabolism. We've already discussed how the first four of these factors can influence a molecule's ability to cross lipid membranes. These principles apply in the same way to absorption of compounds from the GI tract. You should be able to predict how changing pH of the GI tract can influence the absorption of acids and bases. Now we'll go over the influence of surface area, stomach contents, and first-pass metabolism in more detail and finish with some example problems. Surface area is maximized in the GI tract to facilitate the absorption of water and nutrients. This also applies to the absorption of drugs and toxicants. In the intestine, folds, villi, and microvilli create huge area for absorption, giving the intestine a size of approximately 200 square meters, which is about the size of a tennis court. Because the surface area of the intestine is so much larger than all the other areas of the GI tract, the absorption of drugs is usually greatest in the intestine, even if the physiochemical properties of the drug favor absorption in a different area. This idea might seem counterintuitive, but we'll discuss it further in an example at the end of the video. The presence of food in the GI tract can alter the speed and amount of a drug that is absorbed. Food can influence a number of factors, including gastric emptying time, bile flow, pH, GI motility, and bacterial microflora. Absorption can also be altered by direct interactions between drug and food components, or between drugs and other drugs taken at the same time. For example, grapefruit juice can alter the activity of an enzyme found in the GI epithelium and liver that metabolize many drug compounds. This can have a large impact on the amount of drug that reaches the circulation. In the next slide, you'll see why. Any compound that is absorbed through the GI tract and passes through GI epithelial cells is subject to biochemical modification in these cells by specialized metabolic enzymes. Then, all compounds that cross the GI wall pass into the portal vein, which travels directly to the liver. In the liver, these compounds are subjected to an even greater amount of metabolizing enzymes, which can modify the compounds and eliminate some through the bile duct. This pre-systemic metabolism is called the first-pass effect. First-pass metabolism can greatly alter the amount of a compound that reaches the systemic circulation. The amount of a drug that reaches the systemic circulation is termed the bioavailability. Drugs can have low bioavailability because they are not absorbed well from the GI tract, or because they are extensively metabolized during first-pass metabolism. Drugs with low bioavailability are typically not given orally. Don't worry about the specifics of the metabolizing enzymes in the liver and the GI tract right now. We'll discuss them in more detail when we go over metabolism of drugs and toxicants. The oral route of administration is the most common for human drugs. Because many of you may be interested in pharmacology and toxicology research, we'll discuss one additional route of administration that's very uncommon in humans, but commonly used in research. Intraperitoneal injections into the abdominal cavity are absorbed by veins in the mesentery and then gather in the portal vein, which goes directly to the liver and undergoes first-pass metabolism. This type of administration is commonly used in research animals because it's easier to administer a consistent dose. In humans, this type of administration is also used for highly toxic chemotherapeutic drugs to treat cancers within the abdominal cavity. This allows a high dose to be given at the site of the cancer before it's absorbed without high toxicity once it reaches the bloodstream, because the drug will go through first-pass metabolism and some will be eliminated before reaching the circulation. Now let's look at aspirin as an example of how physicochemical properties will affect absorption. An aspirin is given orally. It has a pKa of 3.4. The stomach has a pH of 2. Using the Henderson-Hasselbalch equation, we can determine what fraction of the drug is non-ionized in the stomach. Taking the pKa minus the pH, we get 1.4. And if we take the antilog of 1.4, which is 25, we can determine that the ratio of non-ionized to ionized drug is 25:1. Knowing that a non-ionized drug is more easily absorbed across biological membranes, we can assume that absorption from the stomach is favored. Now if we look at aspirin absorption in the intestine, which has a pH of 6, we see that the ratio of non-ionized to ionized is 1 to 500, suggesting that absorption is not favored. However, this doesn't take into account the effect of having a large surface area for absorption in the intestine. To get a better idea of how surface area will affect drug absorption, let's look at a similar example. Let's compare it to the absorption of salicylic acid, a metabolite of aspirin, from both the stomach and the intestine. The pKa of salicylic acid is 3, and this example uses a stomach pH of 3. In the stomach, the compound will be 50% unionized, so absorption is favored. Now, if we compare that to the intestine where the pH is 6, a much smaller fraction of the salicylic acid will be unionized, implying that absorption is not favored in the intestine. However, if you take a closer look at this graph showing the percent of a dose remaining in the GI tract over time, you can see that absorption from the intestine is much quicker than absorption from the stomach. Although pH will influence absorption from different regions of the GI tract, Surface area is still the most important factor. In this example, the large surface area of the intestine allows the salicylic acid to be absorbed more quickly than in the stomach, despite the fact that it is less favorable in that location due to its physicochemical properties. The bioavailability is defined as the percent of administered drug that reaches the systemic circulation, the circulating blood. Bioavailability can range from 0 to 100% depending on how well it is absorbed. Because IV administration directly enters the systemic circulation, it will have a bioavailability of 100% for all compounds. The bioavailability of other routes of administration is typically less than 100% and can be determined by comparing plasma concentrations following administration to those obtained from IV administration. For a drug taken orally, the bioavailability will be impacted by absorption from the GI tract and the first-pass effect. Because bioavailability is the percentage of a drug that reaches the systemic circulation, it is not affected by things that occur after the drug reaches the circulation, such as binding to plasma proteins. These are the important basic concepts of drug and toxicant absorption. Next, we will cover the distribution of drugs and toxicants in the body. To test your understanding of this video, you should be able to answer the following questions.